Good evening to all the NEET aspirants. So, here we are with biological classification session 2. Okay. In our previous session, what did we discuss beta? Living world, taxonomic categories, and we have exercised few objective, and then we have started with biological classification. We have dealt with complete fundamental insights of biological classification, and now we are fundamental insights and kingdom systems of classification is done. Okay. Two of you sit here, ma, in this row. Siddharth. Yeah, fine. Kunal, you sit there. Okay, fine. So, now in our previous class we have dealt with kingdom systems of classification. Tell me who introduced the first scientific method of classification? Aristotle. Okay. And tell me who gave us two kingdom system of classification? Linnaeus. What are the two kingdoms he has introduced? Plantae and Animalia. Okay. What are the criteria for his classification? Criteria: cell wall, mode of nutrition, response to external stimuli, locomotion, and contractile vacuoles. Okay. Of amongst these 5 criteria, tell me which criteria is majorly considered. Cell wall is the major criteria for two kingdom system of classification. What are the major drawbacks of two kingdom system of classification? Prokaryotic and eukaryotic organisms are placed under one kingdom. Unicellular, multicellular placed under one kingdom. Photosynthetic, non-photosynthetic all are placed under a single kingdom. Okay. What are the examples? Tell me like examples of prokaryotic and eukaryotic. Prokaryotic bacteria placed along with eukaryotic organism. Algae. Okay. Unicellular, multicellular, what example? Chlamydomonas and Spirogyra. Tell me autotrophic and heterotrophic organisms. Green algae and fungi. Okay. So, till here it is fine now beta. 3 kingdom system of classification is introduced by Ernst Haeckel. What is the new kingdom he has introduced? Protista. 4 king. Very good. 4 kingdom system of classification is given by Copland. What is the new kingdom he has introduced? Monera. 5 kingdom by R.H. Whittaker. New kingdom introduced in 5 kingdom is fungi. Okay. What are the criteria for 5 kingdom system of classification? Cell structure, mode of body organization or thallus organization, mode of nutrition, reproduction, phylogenetic relationships. These are the 5 criteria. Amongst these 5 criteria, tell me which criteria is widely considered or majorly accepted? Mode of nutrition. Tell me amongst these 5 criteria, which criteria is because of which criteria 5 kingdom system of classification is widely accepted. Amongst these 5 criteria, because of which criteria 5 kingdom system of classification is widely accepted? Phylogenetic relationships, as he has considered evolutionary relationships, that is the criteria which made 5 kingdom system of classification widely accepted. So, this is all about 5 kingdom systems of classification beta. In 5 kingdom system of classification, there is a comparative paragraph below the table in your NCERT, wherein he compares in between 2 kingdom system of classification and 5 kingdom system of classification. So, 5 kingdom system of classification has put together few unicellular organisms. What are they, under which kingdom? What are those unicellular organisms I told you yesterday? Chlamydomonas, chlorella is placed along with protozoans and amoeba under the kingdom Protista. Okay. And tell me 6 kingdom system of classification is given by? Think, refer the notes. 6 kingdom system of classification is given by? Online students, you can also answer ma. 6 kingdom system of classification is given by Carl Woese. Carl Woese, 6 kingdom or 3 domain system of classification is given by Carl Woese. According to Carl Woese, he said that there is a common ancestor. That common ancestor is progenote. It has 16 as rRNA. What are the 3 domains that are introduced by him? 3 domains? Archaea, bacteria, eukarya. Under archaea domain we have which kingdom? Archaebacteria. Under bacteria domain we have what kingdom? Eubacteria. Only one kingdom under archaea. Under one domain archaea we have Archaebacteria, that is only one kingdom. Under bacteria we have only one kingdom, that is Eubacteria. Under Eukarya we have 4 kingdoms. What are they? Fungi, Protista, Fungi, Plantae and Animalia. As such you got 6 kingdoms, right? 3 domains, 6 kingdoms. Common ancestor is progenode. It has 16S rRNA given by Calvus. All these are kingdom systems of classification. In this we are going to learn the 5 kingdoms in detail from 5 kingdom system of classification: Monera, Protista, Fungi, Plantae and Animalia. Okay. So today we will complete Monera and Protista, and at the end we will exercise the objective also. NEET PYQs also we will exercise. Okay. So stay tuned, ma. So first you have to know what are the general characteristics of the kingdom Monera. All are prokaryotic. Prokaryotic means what? They will not have a nuclear membrane. Let us assume this as a cell where the genetic material is not scattered all across the cytoplasm, but that is placed at a position which is called as nucleoid. This is not surrounded by a nuclear membrane. Okay. So there is no nuclear membrane, there are no membrane bound organelles, nucleoid is present. These are the major features of what? Prokaryotic cell. We have learnt in Cell the Unit of Life, right? And next, histone proteins are absent, okay, and membrane bound organelles are absent. And there is, based on the cell wall composition, we have different types of monerans, okay. Of course, bacteria are the sole members of kingdom Monera. But then based on the cell wall composition we have different classification of bacteria. Eubacteria, cell wall is composed of what beta? Peptidoglycan. Archaebacteria, cell wall is composed of polysaccharides and sugars. And Mycoplasma, there is no cell wall. Mycoplasma doesn't have cell wall. Actinomycetes, cell wall is composed of mycolic acid, okay. And nucleoid, as it is non-membrane bound, this is also called as incipient nucleus, which is composed of double-stranded circular DNA, RNA and polyamides. Double-stranded circular DNA, RNA and polyamides. These are the general characteristic features of the kingdom Monera. Any doubt till here, beta? No, right? Yeah. Next, no issues. I will forward this PDF. You can take down every point in detail. I will forward the PDF, but right now focus on the class. So there is something that we have to connect with the Cell the Unit of Life and learn. Once let us recall the basic structural composition of prokaryotic cell. Okay, prokaryotic cell has an outer cell envelope, yes or no? Cell envelope has 3 layers. Outer is glycocalyx, next to that we have cell wall, and next to that we have cell membrane. Okay, cell membrane will have few extensions into the cytoplasm called as mesosomes. Remembered? Recalled? Yeah. And we have 2 genetic material in the cytoplasm. One is called as genomic DNA and the other is called as non-genomic DNA. Okay, non-genomic DNA is nothing but plasmid DNA, and we have ribosomes also, cytosolic ribosomes, and the ribosomes which are attached to the cell membrane as well. 2 ribosomes we will, in 2 locations we can see the ribosomes. What type of ribosomes are present beta? 70S ribosomes are present. And here near the nucleoid we have what? Double-stranded circular DNA. Okay, genetic material is what? Double-stranded circular DNA. Basic gist and framework, clear, right? And as we have discussed when we are learning the 5 kingdom system of classification, that monerans will show high metabolic diversity. Rajiv, following with a, yeah, monerans will show high metabolic diversity. High metabolic diversity ante enti? What is high metabolic diversity?
Both heterotrophic, autotrophic,
both in the nutrition and in respiration. They are following diverse pathways. For example, in nutrition we have autotrophic, heterotrophic. Under autotrophic also we have types. Yeah, heterotrophic also we have types. Respiration also we have aerobic, anaerobic. Under that again obligate, facultative we have. As such, which kingdom amongst the 5 kingdom system of classification will show maximum metabolic diversity? Then our answer should be what? Kingdom Monera. Clear, right? Next, nutrition. It is both, as I said you, autotrophic, heterotrophic. Under autotrophic we again have photosynthetic, chemosynthetic. Heterotrophic we have saprophytic, symbiotic and parasitic. Next. Yeah, respiration also the same beta. We have obligate aerobes, facultative anaerobes. It is very simple. Most of the students will be confused in between the terms of facultative anaerobe and facultative aerobe. Facultative, you replace this term with sometimes. Okay. Obligate aerobe means it will oxidize the food material, whichever it is taking, it will oxidize the food material only in the oxygenic conditions. Those are called as obligate aerobes. Facultative anaerobes means sometimes it is acting as anaerobes. Okay. Wherein sometimes it is acting as anaerobes means what? Most of the times it is aerobic only in the presence of oxygen. Whenever oxygen is not present, it will also act as anaerobe. Same the case with facultative aerobe. Most of the times it is respiring through in anaerobic conditions. In anaerobic conditions, whenever oxygen is there also it will respire. Understood, ma? Next, reproduction. Reproduction is both by asexual and sexual methods. Sexual as it includes only one genome, right? So, we will call it as gene recombination also, or sexual recombination also we will call it as. We have three types. What are they, Nana? Conjugation, transformation and transduction. Till here it is clear, right? Yeah. Bacterial appendages. We have motile and non-motile bacterial appendages. Motile includes only flagella. Remember, cilia is present only in the ciliated protozoans. Example, Paramecia, they come under Protista. Those are what? Unicellular eukaryotes. Okay. But in Monera, the motile structures, only motile structures we have is what? Flagella. Flagella also will show what? 9+0 condition. Yes or no? Okay. And non-motile structures, we have Pili and Fimbriae. Okay. Now classification you see, based on the presence or absence of the cell wall, habitat, type of cell wall, we have different types: Archaebacteria, Eubacteria, Mycoplasma and Actinomycetes. Let me test your memory. Okay, we have learned the cell wall composition just now. Tell me, peptidoglycan is present where? In which? Yeah. Polysaccharides and lipids? Archaebacteria. No cell wall?
Mycoplasma.
And tell me, what is the cell wall composition of Actinomycetes? Mycolic acid. Okay, mycolic acid. Archaebacteria, look into the general characteristic first. Most primitive monerans occur in extreme harsh habitats. Archaebacteria, they occur in extreme harsh habitats. Based on the habitat they are surviving in, we have three types of Archaebacteria. Okay, what are they? Halophiles, thermoacidophiles and methanogens. Halophiles, they grow in extreme salty areas. Thermoacidophils, they occur in hot springs and they will tolerate high temperature, high acidity and low pH. Low pH means acidic environments. Okay. And methanogens, they occur in marshy areas. They will also occur in the gut of the ruminants. Till here it is clear. Any doubt anywhere? Ruminants, cow and buffalo. Okay. Ruminants. So, examples are important again. Everywhere examples are important. Biological classification, morphology. Without examples, don't expect a question. Okay. Pakka, with examples you will get a question for sure. So, halophiles, examples include Halobacterium and Halococcus. Thermoacidophils, Thermoplasma and Sulfolobus. Methanogens, Methanococcus and Methanobacterium. Okay. Methanogens, as I told you, they occur in the gut of the ruminants, wherein carbon dioxide, methanol and formic acid, whichever they are taking, high rich cellulosic food like cow and buffalo, they feed on what? Cellulosic food like grasses and all, right. So, they will be converted into what? Methane, and that is used as what? Biogas. In microbes in human welfare we have learned, right, production of biogas, how gobar gas is prepared, that is from this only, methanogens. Methanobacteria plays crucial role in preparation of what? Methane, which is used as biogas. Clear, right? Any doubt anywhere? No? Yeah. Next. Here you see in eubacteria once again that just now we have recalled about structures, right? What is the only motile structure? Flagella. And where do you find ribosomes? attaching to cell membrane and in cytoplasm. What type of ribosomes you will find? 70S ribosomes. And you have cell wall is composed of murein, that is peptidoglycan, and cell membrane has extensions called as mesosomes. Okay, ribosomes are purely involved in protein synthesis.
It's a typical bacteria.
Based on the shape, bacteria has 4 categories. Again, bacteria has 4 categories based on shape. If it is circular, cocci, circular cocci, broad shaped, spherical or circular cocci, rod shaped, bacilli, spiral shaped, comma shaped, vibrio. Okay. So, amongst all this, which is most pathogenic? Amongst all this, which is most pathogenic? Bacilli is most common form of bacteria which is pathogenic. Remember that. Okay. And then metabolic diversity, as I told you, anabolism includes nutrition, catabolism includes respiration. Same the thing, flow chart, whichever I have explained just now. But then you have to have a clear idea on photoautotrophic and chemoautotrophic nutrition. See beta, the name itself is saying photoautotrophic means the energy utilized will be sunlight. Chemoautotrophic, the energy utilized is in a chemical substance form, and that is ATP. Here ATP is utilized as energy, chemical energy, okay, for preparation of food. And here in photoautotrophic nutrition, the electron donor is water. Okay, if it is conducting oxygenic photosynthesis. If at all it is conducting anoxygenic photosynthesis, then the electron donor will be H2S. Whereas here inorganic electron donors will be there based on the nutrients they are preparing. For example, nitrogen metabolism, their nitrates will be there. Ferrous bacteria where iron will be there. Hydrogenomonas will have electron donors as hydrogen. Like that, inorganic electron donors will be there in the chemoautotrophic nutrition. So, these bacteria are highly helpful in recycling the nutrients in the environment like nitrogen, phosphorus, ferrous and sulfur. One example I can give you, nitrifying bacteria from nitrogen metabolism, wherein ammonia is first converted to nitrates. Ammonia is first converted to nitrates by nitrosomonas and nitrosococcus. Nitrates to nitrites by nitrobacter and nitrocystis. Remember that. And here examples for photoautotrophic nutrition includes chloronema and chlorobium. Clear, right? What you have to remember with respect to photoautotrophic and chemoautotrophic beta? Energy utilized in both the areas, electron donors, amongst these two which is highly helpful in recycling nutrients. One example from nitrogen metabolism, here also examples chloronema and chlorobium. Fine, right? Clear? Yeah, next. Now we will see heterotrophic nutrition. Saprophytic mode of nutrition, how it will be is, bacteria in their close proximity wherever food is available, it will release out the enzymes to degrade that complex food material. Once that complex food material is oxidized to simpler substances, then they will be consumed by this bacteria back. Understood everyone? Okay. So, here majority of them will be heterotrophs. What they will do? They will secrete the digestive enzymes, degrade that complex material, and the simpler substances will then be absorbed by the bacteria. Clear, right? Next. Here you see symbiotic mode of nutrition. Symbiotic mode of nutrition, best example we can give is nitrogen fixation in leguminous plants by Rhizobium leguminosarum. Both of them are symbiotic association, where Rhizobium is an obligate symbiont here, okay, which will fix the nitrogen in leguminous plants. Okay. And then parasitic mode of nutrition, examples include E. coli and Salmonella. Clear? Any doubts anywhere? Both autotrophic and heterotrophic mode of nutrition is clear to all, right? Yeah, with the examples. Next. Respiration you see beta. Aerobic and anaerobic respiration. Aerobic respiration we have obligate aerobes and facultative anaerobes, means most of the time they are aerobic, sometimes they act as anaerobes in the absence of oxygen. Examples are very important. Obligate aerobe example is Bacillus subtilis. Facultative anaerobes, Pseudomonas, and here obligate aerobes, facultative aerobes. Obligate aerobe example Clostridium. Facultative aerobes means sometimes they are acting as aerobe in the presence of oxygen. Example includes Chlorobium. So, most of the photosynthetic bacteria will act as what? Facultative anaerobes only. All the points are highlighted here. Understood, right? Any doubt anywhere, ma? Yeah. Next. Here you see the standard solid example which is given in our NCERT under eubacteria is cyanobacteria. Okay. Once go through the diagram, I'll just have some water. Yeah, so here you see ma, cyanobacteria. It can be unicellular, filamentous or colonial, but here we are looking into a filamentous cyanobacteria. Cyanobacteria is also called as what? Blue-green algae. Why cyanobacteria is prokaryotic? If that question comes, what will be your answer? Because devoid of nuclear membrane. Devoid of nuclear membrane, it doesn't have membrane bound organelles. Okay.
No histone protein.
No histone proteins, that will be the characteristics. That is why cyanobacteria is prokaryotic, wherein its body is covered by a gelatinous sheath. Okay, body is covered by a gelatinous sheath, and cyanobacteria will have two types of cells beta. One is vegetative cell and the second type is heterocyst. Vegetative cells and heterocyst. Vegetative cells will have both photosystem 1 and photosystem 2. We all have learned from photosynthesis in higher plants. What is the role of photosystems? Photosystems will be having accessory pigments and core pigments where they will trap the sunlight, okay, and they will yes, they will trap the sunlight and they will fix the food material, okay, and they will fix the energy. Fine? Yes, yes. So, PS1 and PS2 will be present in vegetative cells, and then it has chlorophyll A, the pigment which is similar to the higher plants. Chlorophyll A is present. Electron donor is H2O. When electron donor is H2O, now you have to tell me whether it is conducting oxygenic photosynthesis or oxygenic photosynthesis. Remember, each and every point is important, okay. And here you see heterocyst. Heterocyst, these are the specialized cells where you see in the blue-green algae. These specialized cells will have only one photosystem. PS1 will be there, and this generates only ATP. Okay, because photosystem 1 is there means it will generate only ATP. If PS2 is there, then NADPH will be formed. Photolysis of water also will happen. This is used. This PS1 which generates ATP, right? This ATP is utilized for nitrogen fixation. Remember, heterocyst walls are very thick with thick wall depositions which is impervious for gaseous exchange. Okay, this is impervious for gaseous exchange. We can expect a multi-statement question on this explanation, so pay utmost attention and listen. Two types of cells we have come across. One is vegetative cell, second is heterocyst, right Nana? This wall layer is very tough, this is impervious for gaseous exchange, okay. So, inside this only PS1 is present, ATP which is produced here is utilized for nitrogen fixation. For nitrogen fixation, the enzyme required is nitrogenase. Okay, for nitrogen fixation here, what enzyme we require? Nitrogenase is the enzyme. This nitrogenase will function only in the anoxygenic conditions. Understood? Yes or no? Nitrogenase will function where? Only in the anoxygenic conditions. In the oxygenic conditions, nitrogenase will be inactive. So, that is the reason, though vegetative cells are releasing out oxygen, it is conducting oxygenic photosynthesis, the effect will not be on nitrogenase because the wall layer of heterocyst is thick, impervious for gaseous exchange. Understood my point, everyone? Yeah. Now, the membranes, cell membrane will have few invaginations inner to the cytoplasm. These are called as chromatophores. There in the prokaryotic cell from chapter Cell the Unit of Life also we have learnt. Those invaginations towards the cytoplasm in the other bacteria are called as mesosomes, but in cyanobacteria they are called as chromatophores because they are colored pigments. Understood? And then If you look into the cytoplasm also, you can see two types of cytoplasm here, peripheral colored chromoplasm and central colorless centroplasm, two types of cytoplasms, right? And then membrane, this heterocyst membrane is impermeable to oxygen. All the points are covered, right? I will give you a gist. Vegetative cells, heterocyst. What are the components present in the vegetative cell? Components present in the heterocyst. Okay? How and where nitrogenase will be active? What are the invaginations of cell membrane? Okay? And what is the cytoplasm differentiation here? Peripheral is chromoplasm, central is centroplasm. Okay? Clear, right? Cyanobacteria structural description is clear to everyone? Yeah. Now you tell me what are different types of cyanobacteria. This is the description from your NCERT. What are different types of cyanobacteria and where do you find the cyanobacteria?
Unicellular colony.
That is the description of cyanobacteria. I asked you what are different types of cyanobacteria or BGA. Nostoc, Anabina, Olocera, Oscillatoria. These are different types of cyanobacteria. Okay, so these are unicellular, colonial, filamentous, aquatic or terrestrial algae, and they are photosynthetic autotrophs. They are most primitive organisms to conduct oxygenic photosynthesis. Remember that statement is very important. What are the primitive organisms to conduct oxygenic photosynthesis? Cyanobacteria. Okay, they form algal blooms in polluted water bodies, because of which eutrophication occurs. Okay, algal blooms are formed in the polluted water bodies by cyanobacteria. Best example is Trichodesmium erythraeum, we will come across in the coming slide. Next, heterocysts, these are the special cells to fix the nitrogen. Okay, examples Nostoc and Anabina. You can also take Olocera and Oscillatoria. Next you see. Apart from heterocysts, you will also find some specialized cells called as echinates beta. Echinates are nothing but in between vegetative cells and heterocysts, few cells will get dead. Okay, by losing their centroplasm, chromoplasm, they will get hard and dead. That will lead to fragmentation. Okay, and that fragment will develop into a new individual. As such, it will promote the vegetative propagation. Okay, echinates. See, for example, these are vegetative cells and this is heterocyst. Vegetative cells and heterocyst. Amongst this, let us assume this is the echinate. Very thick and tough wall. By losing their centroplasm and chromoplasm, they get hard. Okay, and this fragment will be break here. Okay, that gives rise to a new fragment of BGA. Understood? So as such, it will promote what? Propagation. Next, as we have discussed, Trichodesmium erythraeum is a marine cyanobacteria which forms red color of Red Sea. Here only I am telling you one more point, that in dinoflagellates, when you are learning, there is one more example called as goniolax. That is dinoflagellate, that is eukaryotic, which causes red tides in the Red Sea. But cyanobacteria example is Trichodesmium erythraeum, which causes red tides in Red Sea. This is marine cyanobacteria, which is prokaryotic. Remember, okay, do not get confused between Trichodesmium erythraeum and goniolax. Clear, right? Yeah. Next, reproduction, asexual reproduction and sexual reproduction. Asexual reproduction under favorable conditions, binary fission occurs. This is the most common mode of asexual reproduction. Under unfavorable conditions, endospores are found. I will tell you, I will give you a brief description on the endospore also now. And sexual recombination is conjugation, transformation and transduction. This is the basic idea on the reproduction of monerans beta. Okay. Now you see. First is binary fission, which is most common. There is no spindle formation here, just karyokinesis and cytokinesis will happen. Karyokinesis, cytokinesis and a single cell forms to two cells. Okay. So this is also called as amitosis, as there is no involvement of spindle fiber formation. Second is endospores. Endospores, outer wall is exosporium, next to that spore coat will be there, and next to that cortex. Most important point, cortex is composed of calcium dipicoic acid. Okay, cortex of this is composed of calcium dipicoic acid, which makes endospore resistant to any environmental condition. Okay, look here, This has what? Endospores will have low water content, they have low metabolic activity. This is, endospores are unharmed during pasteurization, and endospores, they are means of perennation but not reproduction. Endospores are what? Means of perennation but not reproduction. Till here it is clear, Noma? Any doubt anywhere? Yeah. Abdul, any doubt? Binary fission and endospores, clear, right? Yeah. In favorable conditions, whenever favorable conditions resume back, endospore will open up and the bacteria will be released out. The wall layers will open up and the bacteria will release out, of which one endospore will release one bacteria. Remember that. One endospore releases out how many bacteria? One bacteria. Next is sexual recombination. Sexual recombination, this does not involve no gamete formation or gametic fusion here. Okay? There is no gamete formation or gametic fusion, that is why we call it as sexual recombination. First type is transformation, which is proved by Griffith's experiment on the model organism Streptococcus pneumonia. You have to know two strains of Streptococcus pneumonia, beta. One is R strain and the other is S strain. R strain is rough strain, okay, which is unharmful because it has just wavy outline of the cell wall. But smooth strain has a very smooth coat over the bacteria which will protect the bacteria from host immune system. But rough strain of Streptococcus does not have any outer coat. So, this will be easily killed by the host immune system. Understood? So, of R strain and S strain, which will be virulent? S strain, because it has strong smooth outer coat. It can survive in the host immune system. Understood? Yes. So, here you look at the experiment now. First, the mice is injected with a living virulent S strain. As S strain can survive happily in the host, mice will die. Okay. Second, when R strain is injected, R strain will die because of the action of the immune system from the host. Next is heat killed S strain. When S strain is heated, the outer smooth coat is damaged and the mice is alive. Okay, where S strain, when S strain is heated, mice will be alive because of the effect of smooth coat get damaged. As smooth coat gets damaged, mice will be alive. And then living R strain along with dead S strain when it is mixed. Heat killed S strain, when it is mixed with R strain, the mice got killed, which means that there is something that is transformed from heat killed S strain to R strain, which made the R strain virulent. Yes or no? Okay, this made R strain virulent, so mice has dead. So, here you see there is no contact between, here there is no contact between donor and recipient. Is there any contact? No. But then the genetic material is exchanged from the environment of the bacteria. Okay, the genetic material of R strain is exchanged from the S strain from the environment in which it is there. That is called as transformation. So, here comes the, yes, and here comes the concept of competence. Competence is nothing but the ability to pick the DNA from the solution by any bacteria. Okay, ability to pick the DNA from the solution is competence. So, have you understood the transformation experiment? Any doubt anywhere? Again, you will come across this experiment in molecular basis of inheritance. If you are clear here, then there it goes with ease. Okay. Next, conjugation. Conjugation is given by Liederberg and Tatum. Okay. This is the primitive type of DNA transfer. Okay. This is from bacteria to bacteria. So, this is the exchange of genetic material in between two bacteria, which is explained with F factor. As bacteria is having non-genomic DNA or plasmid DNA, we have discussed already. So, that plasmid DNA will have n number of factors beta, like F factor, Ti factor, based on the types of factors it has. If that plasmid DNA is having F factor, then we will call it as what? F plus strain. Okay. This F plus strain will be attached with F minus strain, okay, of other bacteria where F factor is absent in that plasmid DNA. These two bacteria will come to close proximity by a conjugation tube. Conjugation tube is also called as sex pili or true pili. Okay, understood? So, the F factor is present, this is called as what? F plus strain is connected to other bacteria in which F factor is absent. So, this is called as F minus strain. These are connected by what? A conjugation tube. Now, the F factor is not completely lysed over here. Some part of the F factor will be lysed and this is transferred to F minus strain. Okay. Into where? Plasmid DNA of F minus strain. So, at the end what products you will get? F plus, F plus. Okay. Sometimes the transfer of F factor will be from genomic DNA to the plasmid DNA. Genomic DNA of F plus strain to the plasmid DNA of F minus strain. Then that is called as HFR strain. Those bacteria which has F factor in the genomic DNA is called as what? HFR strain, where F factor is transferred from the genomic DNA to plasmid DNA. Okay. So, here at the end what products you will be getting? HFR and F plus. HFR and F plus. Clear? Any doubt anywhere? No, right? Yeah. Next. Transduction. Transduction is given by what? Zinder and Lederberg. Zinder and Lederberg has proposed transduction. Model organism is Salmonella typhimurium, wherein the genetic material is being transferred through a bacteriophage. Okay. Double stranded circular DNA. Don't get excited, huh? So, the genetic material is transferred from where? Bacteriophage to bacteria. The transfer of genetic material through bacteriophage is called as what? Transduction. Okay. Thank you, Bharat. Continue with the class. Next, useful bacteria and harmful bacteria. Okay. Useful bacteria, if you see, we have many examples: making of curd from milk, lactobacillus; production of antibiotics, Bacillus subtilis, and the antibiotic is subtilin; nitrogen fixation in leguminous roots, Rhizobium and Frankia. Next, here you see harmful bacteria includes cholera caused by Vibrio cholerae, typhoid, Salmonella typhi, tetanus, Clostridium tetani, citrus canker by Xanthomonas citri. This is all about the reproduction part beta. So, asexual reproduction we have learned, binary fission, which is the most common mode of reproduction under favorable conditions. During unfavorable conditions, endospores. Sexual recombination, 3 modes we have exercised. What are they? Transformation, conjugation, transduction. Till here it is clear, right? Next, mycoplasma, which is wall-less bacteria called as jokers of plant kingdom. Okay, these are the smallest living cells called as jokers of plant kingdom, and they are pleomorphic. They can change their shape based on the environment they go to. They don't have cell wall, pleomorphic. They are facultative anaerobes. Okay, facultative anaerobes means what? Abdul?
Sometimes anaerobes.
Most of the time they are aerobic, but whenever there are anoxygenic conditions, they are acting as anaerobes. Mostly they are pathogenic to both animals and plants. Fine, right? This is the description of mycoplasma. Fine? Any doubt anywhere? Next.
They are not in plant kingdom, right?
No, they are prokaryotic cells. I am saying they will cause diseases. They will cause disease both to plants and animals.
Then why are they called jokers of plant?
Jokers of plant kingdom because they frequently change their shape. Okay. Now you see, examples include now it is clear, right ma? Witch's broom, pleuropneumonia, mycoplasma, urethritis. These are the diseases that are caused by mycoplasma. Next, actinomycetes, branched filamentous bacteria. Actinomycetes, this should be focused only by EAP set students. NEET students, you will not get question from actinomycetes, but then listen. Branched filamentous bacteria, they will radiate out colonies in culture. Cell wall is made up of what? Mycolic acid. Mostly they are saprophytic and decomposers. Any doubt till here? No, right? Yeah. Next. Here you see parasites. Examples for parasites from mycoplasma includes Mycobacterium and Corynebacterium. Antibiotics include streptomyces. Okay. So till here the kingdom Monera is done. Once let's have a brief discussion on the kingdom Monera. We will exercise few objective and then we will move to kingdom Protista if possible in this session or in coming session. So from kingdom Monera beginning we have learned about general characteristics, yes or no? What are the general characteristics of kingdom Monera? Prokaryotic, single circular, double stranded circular DNA. There is no nuclear and membrane, nucleoid will be there, no histone proteins, okay, and
plasmids are present.
Plasmid DNA, genomic DNA, non-genomic DNA is present. Ribosomes are of what type? 70S ribosomes. Rigid cell wall. Cell envelope has 3 layers. What are they? Outer glycocalyx. Next to that cell wall. Cell wall is made up of peptidoglycan. Next to that we have cell membrane. Cell membrane will have few invaginations towards the cytoplasm. Mesosomes. Okay. And what are the positions of the ribosomes? Cytosolic ribosomes are there. Those which are attached to cell membrane also. What type of ribosomes we see? 70S ribosomes. What is the only function of ribosomes? Protein synthesis. Okay. Genomic DNA and non-genomic DNA. Non-genomic DNA also called as plasmid DNA. Okay. Next, it shows extensive metabolic diversity both in nutrition and in respiration. In nutrition, photoautotrophic bacteria will have what energy? Sunlight as energy. Chemoautotrophic? ATP as energy. Here what will be the electron donor? In photoautotrophic?
Inorganic.
Photoautotrophic H2O or H2S. H2O if it is oxygenic photosynthesis, H2S it is if it is anoxygenic photosynthesis. And here in chemoautotrophic bacteria, what will be the electron donors? Inorganic components like nitrogen, phosphorus, sulfur and all. And here in the anoxygenic photosynthesis, in chemoautotrophic photosynthesis, it is highly useful for what? Recycling nutrients. Best example we have taken nitrogen metabolism. Photoautotrophic also 2 examples we took. What are they? Chlorobium and chloronema. Clear? Next, heterotrophic mode we have learned saprophytic, symbiotic and parasitic mode of nutrition. And then respiration, obligate aerobes, facultative anaerobes, obligate anaerobes, facultative aerobes. Clear till here? Next about reproduction. In reproduction we have learned about binary fission which occurs in favorable condition, and then about endospores which is in unfavorable condition. Sexual recombination includes conjugation, transformation, transduction. Transformation is given by Griffith. Model organism Streptococcus pneumonia. Okay. Genetic material is exchanged from
nearby medium.
Environment, nearby environment. Conjugation given by very good Rajiv, Liederberg and Tatum. Okay, that is the exchange between 2 bacteria, genetic material, where F factor is exchanged from either plasmid DNA or genomic DNA to the plasmid DNA of recipient. Okay, and then we have learnt about transduction. Model organism
Bacteriophage.
Salmonella typhi. Okay, given by Liederberg and Zinder. Okay, Zinder and Liederberg. Okay, and then we have exercised about useful bacteria and harmful bacteria. We will now exercise few objective. Have 10 minutes of time. Yeah, look at this. Just 2 minutes. Yeah, direct chart. Sure?
Yes. Wait.
If it is not, now you see. Cell type here under Monera will be prokaryotic. Cell wall? Cell wall is present. And 1 and 2 are done, right? Under 3, Animalia, nuclear membrane is present. Body organization of fungi is multicellular, loose tissue type of body organization. Now you tell me, what is the right option? Prokaryotic, 2 will be present multicellular. That is about Protista, but then yeah, do this question. The domain archaea is not characterized by linear chromosomes. The domain archaea is not characterized by pseudopeptidoglycan in the cell wall. Yes, it has pseudopeptidoglycan. Introns are present in some genes. Yeah, this I have to explain you. This concept is in connection with Malvaivita. See. So, from 6 kingdom system of classification Calvus, we have 3 domains, right? Archaea, bacteria and eukarya. In these domains, bacteria and eukarya, you have the bacteria doesn't have the split gene arrangement. It just have only expressed sequences called as exons. Okay, whereas archaea and eukarya, these two will have split gene arrangement of DNA, wherein it has introns, exons, introns, exons. The introns are the unexpressed parts of the DNA, okay, which are inactive parts of the DNA. Okay, this split gene arrangement is seen where? In the Archaebacteria and Eubacteria. Okay, clear, right? So, introns arrangement are seen. Yes, methanogenesis is as unique metabolism. Yes, this is considered as unique metabolism. Linear chromosomes and pseudopeptidoglycan in the cell wall. Yes, option B will be the right answer over here. Okay, fine. Next. Look into this. Consider the following regarding the fact that cyanobacteria are now kept in Monera and not in Plantae.
Option
D. Okay, they are prokaryotes, correct? The cell wall of cyanobacteria has peptidoglycan, correct? They can fix the atmospheric nitrogen, correct? So, option D will be the right answer. This is about Protista, fine. So, in our next session, we will discuss completely about Protista and the questions from Protista. Fungi also will start it better. So, that is all for today's session. Go through the complete kingdom of Monera, practice all questions. You can expect the questionnaire in connection with what? Cell, the unit of life, prokaryotic cells. You keep that as reference, connect the concepts and learn. And as we have discussed the split gene arrangement from Archaebacteria and Eukaryotes. That also you just go through once, fine. Refer and practice all the questions from our package. DPP NCERT beginners should be done by tomorrow, okay. Yeah, that is all for today's session, beta. Thank you.